Good afternoon, everyone. My name is David Yáñez from the Spanish Research Council. I guess some of you know me already. I'm coordinating Work Package 3 and involved in other tasks in other work packages. And my main area of work is my main area of expertise is ruminant nutrition, and in particular the optimization of the rumen fermentation process in the animal with a special focus on methane emissions. So the idea for the next 20 minutes or so is to provide a very general overview of the main sources of greenhouse gases emissions related to livestock production, and to present some examples of the link between the biology or the microbiology involved in these emissions, and how we can link that to the development of different emission factors to be able to calculate or to estimate emissions from these different sources. So as I said, it's going to be a very general overview, but it's an exercise of linking biology with numbers that I think it's important in the context of pathways. So see if I can, yeah. So, so we start with this general graph that some of you may know, which is just a graph showing the different sources of greenhouse gases emissions for the different sectors, and within each sector, different activities. In our case, in agriculture, in particular livestock, the main two sources of emissions are derived from the physiology of the animal and the digestion of the degradation of manure, which generate mainly methane, and also the activity occurring in the soils that are used to grow feed for the animals that mainly produce nitrous oxide. But there are other emissions, CO2 emissions related to the land used to grow feed, mainly through deforestation, and then other sources of CO2 through the use of electricity, heating, transportation that are considered in other sectors. These three gases, carbon dioxide, methane, and nitrous oxide, we know they're different, not only in the global warming potential in relation to CO2 as the reference, so how much they contribute to heat the atmosphere, but also the time they spend in the atmosphere, the lifespan. And this is very important in terms of prioritizing different measures, regional, national, or international level. But it's something that we're not going to discuss today. So today I'm going to use like a simple, very general dairy farm as an example. And in very general terms, we can consider the following sources of greenhouse gases emissions. The first one is related to the different inputs, not only feed production, which is important, but also pesticides, fertilizers, machinery, animals, manure, etc. And these, in general, they generate CO2 and N2O. The other category is the animal physiology itself. In ruminants, methane is the main gas produced by enteric fermentation that is occurring in the rumen, and we will see some details later on. Then the third category is related to the management of manures, of feces and urine produced in the farm. They are subject to a different reaction depending on how they are stored and how they are managed, and they generate mainly methane and nitrous oxide as a result of those processes. And then there is another category in which we can include the use of energy for different purposes in the farm, outside the farm, processing of some of the products, transportation, etc., and they generate CO2. So these are, in very general terms, the four main categories that contribute to the emissions of greenhouse gases, but we can also consider another category that can actually reduce those emissions through carbon sequestration. So the soil in the farm has the ability to retain some carbon, and it's always a balance between retention and emission. And when the retention is greater than the emissions, there is carbon stored, and this can be measured. It's quite challenging actually to model it through a range of different systems and climatic conditions. But it's something that it is important to consider. It is a medium long-term a process that can be very quickly reversed as well. So it's a complex process. And this is something that Work Package 5 and Work Package 3 are working to try to improve the estimations in our analysis, but we are not going to be talking about that today. We can consider a more complex system of view of the emissions by, well, the whole thing. When we account for emissions and sequestration and we put everything together, is basically what we call the carbon footprint of a product. In this case, it could be a kilogram of milk. So this graph can be considered in a more complex way with emissions occurring in the farm, what we call direct sources from feed production, animal physiology and manure handling. Then what we call the pre-chain sources that you can see on the left-hand side. And as I said before, any input that comes in the farm, not only material, but animals or organic material as well is considered. And then the indirect sources occurring post-farm. So the whole thing, the whole cycle has to be fully considered, and then that makes the whole analysis quite complex. Today we're going to just use a couple of examples from pre-chain sources and direct sources on the farm, just to illustrate the development of emission factors. We can use these two models as examples of how the different sources of emissions contribute to the emissions of the production of milk in this case. So this is an intensive farm in the US, and this is an extensive grazing farm in New Zealand. As you can see, in both cases, the production of enteric methane from the animal is the main source of emissions, but then the proportional importance of other sources changes from one system to another. So in the grazing system, there's a big chunk coming from the crop and pasture land management, while in the intensive systems, the storage and the management of manure and the pre-chain sources, mainly through feed imported into the farm, are the two main other elements in addition to enteric methane. The question here is how we can accurately calculate or estimate these emissions at not only a farm level, but a region or national level. And if we use the IPCC umbrella, and using again enteric methane as an example that I'm more familiar with, we can use 3 levels of complexity to estimate these emissions. And these levels are called tier 1, tier 2, and tier 3. If we start with a very basic one, tier 1 is basically a system in which a fixed amount of emissions is allocated per animal and year, regardless of the system where the animal is. So it's very simple. So if you know the number of animals in the farm or in the region, then you can easily calculate how much, in this case, how much methane is produced. It's very simple, but it's not very accurate because it doesn't consider many specificities of the farm system. We can go up in another level of complexity, the tier 2, which is the one that is used in most cases, in which we start considering other aspects of the system. In this case, the most relevant is the diet, so the composition of the diet, of the ingredients, and digestibility, and we obtain what we call the YM, which is the emission factor that determines how much of the original energy in the diet is lost as methane in percentage. So, for instance, this would be 4%. So by knowing that, we can apply this percentage to different kind of diets or different livestock categories within different type of farms in each country. So we can better characterize those systems, in this case for methane emissions. And then we can go up to The top level, a tier 3, which is more sophisticated, is normally country specific, using models that use data specific for that country, in which we include more details of the diet, the concentration of the fermentation products in the rumen, and also we can even consider a different type of mitigation strategies. And this is not as usual to have it developed in different countries. So only a few countries have this tier 3 system developed. Right. The more advanced we are in the tier system, so the more complexity we add into the calculation, the better, because we can first better describe the system. So we can include as many specificities of the system as possible to better characterize it. But more importantly, we can capture the potential improvements as a result of implementing new management or innovations, which is basically one of the things we want to do in Pathways. We want to see how much how we change the environmental impact as part of the sustainability of the systems changes depending on what we do in terms of management. But for that, we need to have emission factors that can really capture the peculiarities of the system. And in order to do that, we need to understand the biology which is behind the development of these emission factors. And this is basically what I want to do in the next 10-15 minutes with two examples. The first one is around feed production and the emission of nitrous oxide. I'm not an expert on this area, but I thought it was a good way to illustrate the importance of the microbiology or the biology behind these processes. So we know that nitrous oxide, we can use different kind of fertilizers to provide nutrients to the plants in the soil, and this could be either urea, ammonia, or nitrate. And there are microbes in the soil that can convert this urea and ammonia into nitrate, or nitrate into nitrogen through different processes called nitrification and denitrification. But in a sense, in these processes, N2O is produced and emitted to the atmosphere, and that can be from either synthetic fertilizers or from manure. Basically, what we really want to know is how much nitrogen is lost or is emitted to the atmosphere as N2O per, let's say, 100 kilograms of nitrogen input as fertilizer or as manure. In principle, the IPCC by default tier 1 guidelines used 1% as the default value. So 1% of the nitrogen applied here is lost in N2O, but there are many factors that can influence the activity of this bacteria in the soil related to moisture, pH, temperature, the content of carbon that change the result of the N2O emitted into the atmosphere quite substantially. So just to illustrate that changes, this is one graph from a meta-analysis. This is actually in the Mediterranean area where the emission factor is below 1%, so less than 1% of the input nitrogen in the soil is emitted as nitrous oxide. And we can see the type of fertilizer, whether it's organic or is synthetic, or within organic, whether it's liquid or solid, influences substantially the amount of nitrogen that is lost. Another factor that is important is the application rate. So as you can see here, the more nitrogen that we include per hectare as fertilizer, the percentage that is lost as nitrous oxide is greater. So there is a linear exponential relationship that is also important to consider. And there are many other factors, environmental factors like the carbon content in the soil. So the greater the carbon content, the greater as well the emissions of nitrous oxide, temperature, pH, which is an important attribute of the soil. They can greatly influence the activity of these bacteria and therefore change the amount of nitrous oxide that is produced. So as an example, in the latest 2019 IPCC guidelines, updated the previous one from 2006, we can see this is the emission factor in, I think it's kilograms per 1,000 kilograms, gram per kilograms. Basically, you can see the moisture or the rainfall is an important factor. So whether it's wet or dry determines quite a lot the amount of nitrous oxide that is produced per kilogram of input, whether there is irrigation or not, or whether the fertilizer is organic or synthetic. There is a big change in the amount of nitrous oxide that is emitted. And this is just a kind of simple disaggregation. We can consider more aspects and more peculiarities of the system that can be more accurate in estimating how much nitrous oxide is produced. And this is what we want to, what we aim to do in the project and in the scientific community in general. So the second example is the animal physiology, so the enteric methane production in ruminants. We know that the rumen is colonized by a complex and diverse mix of microbes, prokaryotes and eukaryotes that ferment the feed, the carbohydrates and the protein in the forage and the grass to produce energy and microbial protein for the animal. But in this process, they also generate, among other products, methane. If we could open a little hole here on the left-hand side of the animal, basically what we could see, let's see if we can, I need to, yeah, we could see something like that. It's not, this is just only a portion of the microbes that we can see in the rumen because the little ones cannot be seen in light microscope, but basically we see a mix of microbes colonizing the feed that is ingested by the animal and then producing fermentation products that are used by the animal. So in this case, methane is not produced by the cow, it's produced by the microbes, and the farmer is not feeding cows, it's feeding microbes that in turn are feeding the animal. So when it comes to developing nutritional strategies or strategies to reduce greenhouse emissions, sometimes we have to see the process from the microbiology or the biology perspective to really understand the potential for some of these interventions. How enteric methane changes depending on the diet, for instance. But you can see this graph in which we plot the emission factor, the Ym, which is the proportion of the original energy in the diet that is lost as methane, because methane has energy, in relation to the digestibility of the diet. As you can see, as the digestibility of the ingredient of the diet increases, the amount or the proportional amount of methane that is lost is going down. Also for feeds that are low quality with low digestibility, we lose more energy in terms of methane. So we could classify feeds in different categories like low, medium, and high quality. So the higher the quality of the diet, the less methane is produced. So we can estimate how much methane is going to be produced per animal if we know in detail the quality of the diet. And how do we know that? But basically in the last 10-15 years, there's been a big research effort in the scientific community and in the private sector as well, in measuring methane from a range of different systems, different kind of animals in different countries that end up with models and estimations that can allow us to estimate with some accuracy the amount of methane that will be produced in different systems. And to illustrate that improvement that we have made in the last years, this is the table that was provided by the IPCC guidelines in 2006. So not that long ago, for a tier 2 emission factor. So the medium one, in which if we know the type of animal and the diet, we can estimate how much methane is produced. So if you can see, for dairy cattle, there is a fixed emission factor allocated, so 6.5, which is not, it's just a a fixed one, regardless of the system where the dairy cow is raised. And for beef cattle, it can be either 3 or 6.5, but there were no other options for choosing. So in 2019, because of the data that has been generated on the studies, now we can consider whether the animal is high, medium, or low producing, whether the diet has high, medium, or low digestibility, or the content of fiber, and which will give us an emission factor that better reflect the amount of methane that is produced by the animal. And the same applies for beef cattle, in which, depending on the percentage of forage and the digestibility of that forage, then of the diet, we can then go from very low emission of methane to very high, depending on the type of diet. And there are many other specificities in the guidelines. This is just a general table, but we can really go more into detail to characterize these systems, depending on not only the diet but all the factors related to the animal. So so just to finish, show you a few examples of how we can reduce, in this case, enteric methane emissions through different interventions that can be potentially applied in different systems, using the intensive dairy cattle system we had at the beginning of the presentation. One could be the use of nutritional additives, so compounds that can be specifically used and added into the diet to reduce methane production in the rumen without affecting other parts of the production or other attributes of the digestion. So if we do that, for instance, using the recently approved Bovar 3NOP by DSM, we know that there is a 30% reduction in methane emission, and basically what we're going to do is reduce the carbon footprint, if we don't affect the other ones, by reducing 30% this portion of the emissions. Another option could be, for instance, using byproducts, so alternative feeds from the agro industry, to replace some of the conventional systems. And we know that we can reduce 10-50% of the emissions basically by reducing the associated emissions from the pre-chain part of the whole story. So basically, the nitrous oxide emissions that are produced as a result of growing those feeds that are now being replaced. So that would be another option. And we can, by knowing exactly what ingredients are going to be replaced and how those ingredients are produced, then with the right emission factors, we can actually estimate how much mitigation we're going to achieve. And another, this is a more general concept, which is basically increasing the longevity, and this is very relevant in many production systems. So basically, what we're doing is to have animals that last for longer in the farm. So they have, instead of 2 lactations, let's say 3 or 3.5 lactations. And basically, what this is doing is diluting the emissions of the animal in the growing phase. Because during the first 2 years of life of the animal is eating and growing, but it's not producing milk, but it's emitting greenhouse gases. So if we increase the longevity, then we will cut substantially all of these different sources of emission, because we are basically producing more milk with the same animal. So these are just 3 examples of how we could reduce, in this case, enteric methane, to illustrate the importance of knowing the emission factors as we've seen previously. So very, very quickly, 2 main conclusions. Basically, the different sources of emissions have biology, or in this case, microbiology behind that explains how these emissions are produced, and there are different environmental factors that need to be considered to really understand better how these emissions occur and how we can quantify them. And if we have good emission factors that consider all those elements of the biology, then we can hopefully better describe the systems, so we know on which baseline the different systems operate, and more importantly, we can capture improvements as a result of changes in management and innovation. That is basically what we are going to be doing in parts of this project. And I think, that's all for me. I hope I was within the 20 minutes or 15-20 minutes time.
Great, thank you, David. Are there any questions? We can open the floor for a few questions. I see a hand up. Nikki, go ahead.
Thanks, Danielle. Thanks, David. That was really interesting. I was just wondering how far back the pre-chain sources calculations go. So if, for example, feedlot cattle are being fed what had been food identified for human consumption, and then it's a waste product, so it then comes to be fed to cattle, how far back do the emissions... Are the emissions calculated for that? Is it right back to the growing process for the ingredients of those products?
If you have that information, yes. In most cases, the main challenge or the barrier of having an accurate, and I'm not an expert on life cycle assessment, so if anyone can provide more detail on this, please jump in. The limitation is to have the accurate and real information of the whole system. So if you have that information, you can go back to what you said, to the initial phase of growing the raw materials. Yes.
However, if I can complement your response, David, there is a general rule in life cycle assessment that when it is considered a waste has no impact to produce it. When we throw our rubbish into the rubbish bin, the impact related to the rubbish that I'm bringing in is considered zero. But as we go into farm systems and livestock system, the concept between waste and byproduct or co-product becomes more complex. And in subgroup 1 of task 5.1, we'll try a little bit to have a clearer view of what it has been done and what are we missing in relation to this. If I'm allowed, I have a questions and something to add. In H2O, what is also key is the ratio between the carbon and the nitrogen of residues, manure, and whatever that affects the H2O. So it's the relationship between the two. And regarding the emission, beside the rainfall and water availability, you're very welcome, Nicky. It's also when the rainfall happens with regards to nitrogen applications. So is particularly key for the denitrification process. The question that I have for you, David, is you mentioned longevity with regard to the milk product, dairy. I wonder if you can expand a little bit more on the beef side or the meat production side of things. And broadly speaking, also, for instance, the production of fibers, because we shouldn't forget the sheep and goat, maybe not in Europe, are used to produce fiber with regard to longevity.
Yeah. Well, yeah, it's true, obviously, that there are many, just to comment on the first question, there are many other aspects related to fertilization that need to be considered, and how many applications and how the application and climatic events happen. So obviously that all influence, and that changes depending on the area and the geographic place. But yeah, in terms of the longevity, obviously the dairy system, it's the impact is more obvious, because you can have an animal producing milk for as long as the animal is alive and can get pregnant and produce milk. So the more lactations that you have in the animal, the better, because you are diluting the emissions from the first growing phase of the animal. Your question is about meat production. meat production, obviously longevity of the animal that is going to be slaughtered is not an issue, but it is an issue of the mother. So if you account for that in the system, then that would have obviously an impact on reducing emissions if you have less mothers. But then you know that the meat systems are complex because you can have either animals coming from the dairy systems or the purely beef systems, in which you have mothers just giving birth to calves that will be fattened later on. So depending on where the animal comes from, then the impact is different, and the importance of the longevity is also probably different. I can't see who's...
Okay, yeah, there were two more questions. There was one from Adam and then Robert.
Yes, good afternoon, everyone. I have some suggestions to the presentation. The presentation was very good and very interesting, but I think we have some gaps in this presentation. For instance, in cows and sheep, we have 4 cells in the stomach, and we don't have just microbiological digestion, but also we have enzymatic digestion and absorption of the substances in body of the animal. I think is higher, and we cannot just take into account the microbiological digestion. And I think we have higher microbiological activities and biosequestration in soil than we have on this presentation. And in my point of view is the biosequestration in soil by plants is the first level of the biosequestration. Animals which are eating plants, this is the second stage of biosequestration, and animals which are eating animal products are the third level of the biosequestration, and I think we have to take into account this situation because most carbon from the air is coming to the plant and then to the animals, and just we are losing some carbon to the air during whole cycle of the carbon in atmosphere. Thank you. Sorry for my English, but I think you understand what I would like to say.
Thank you, Alam. Yeah, that's okay. Obviously, in a 15 minutes presentation, it's very difficult to go into all the details that you mentioned, and we can have other if you want on the microbiology aspects. I think they are equally important. Regardless of whether it's the soil of the rumen, but yeah, they all have their peculiarities, and that was the point of the presentation, trying to highlight some of the specific aspects of these microbial processes. So if we better understand those processes, then it will be easier to derive accurate emission factors and eventually develop mitigation strategies.
I think I would just to add to one thing that I think the methodology of the emission or footprint of carbon in animal production is too high. I think we have some gaps or misunderstanding in the methodology, and is the footprint and emission from the animal production is too high from my point of view. Thank you.
Okay. Yeah, I respect that opinion. I'm sure that we can improve. When I say I mean the scientific community or the international community. We can improve the accuracy of our estimations, but there are quite a few studies already published that can support some of the emission factors that are officially used. But that's the purpose of one of the work packages in this project, is to improve our estimation factors and to try to be more accurate and to better reflect the peculiarities of the systems. That's why for many of the reasons we are here in this project.
Okay, can we end on Robert then?
Thank you. Hi, David. Hi, all. Very interesting presentation. Just my question is connected to Adam's question. I'm just wondering what impact is the impact of grazing management in terms of pasture for whole circle of greenhouse gas emission, and also in this terms, what is the connection between carbon sequestration in the soil and greenhouse gas emission, and especially methane emission? Did you include this in your studies before, or do you know? Are you aware of some recommendations for grazing and pasture for greenhouse gas emission?
Yeah, that's a very broad question, Robert. Thank you. But I'm not an expert on grazing. Yeah, there are recommendations and studies trying to link the management of grazing and the amount of carbon that can be stored in soils. But as I said before, these changes from one location to another. So depending on the kind of soil and the grass and the system that you are operating with. But in general, I could say that if in a well-managed grassland system, you can increase the amount of carbon that is stored in the soil until an equilibrium is reached. But having said that, then the equilibrium can be broken very quickly and reversed. So yeah, it's something that I don't think is fully developed to be implemented in the models in terms of accounting accurately for the emissions. But I think it's something that is being worked, and there are systems that are more advanced in these calculations, and others, like in some Mediterranean areas and other systems where there is not so much data on the carbon stored in the soils, that need to have more work to really get a better estimation or better modelization of those processes.
May I?
Thank you.
Yes, but we'll really need to.
Okay, just one sentence. Yes, very shortly. I think the biosequestration is not just in soil; it's in body of animals and in the mass of plants. This is also the biosequestration, and in plants we have just biosequestration for a short time. In body of animals, in a longer time. Thank you. Well,
the time frame is it can change, because if it is a beef or poultry, then that time is not alone, and that goes into human consumption and the whole chain. But yeah, of course there is. But this is part of the carbon cycle, of course.
All right. Thank you so much, David, for your presentation and for our questions. We'll convene again next month. So if there's any suggestions on additional topics or presentations, don't hesitate to get in touch with me. Thank you, and have a great rest of your day.
Thank you.
Thank you.
The EB meeting will not take place. Thank you, everyone.
Thank you.
Thank you.
Ciao.
Thanks. Bye.
Thank you. Bye-bye.
Thanks.